gated channel will be located under the terminal knobs. All right, let me emphasize the structure of the nerve cell's axon. I'm not sure that I emphasized that before, but read up on this again. All right, let's look at the axon. The axon is one usually long extension of the nerve cell's cytoplasm. You can look at it that way. It is the way by which a nerve cell sends an impulse away from its body toward another cell, usually a nerve cell, a muscle cell, or gland cell. Now, at the end of the axon will form branches. All right, this is called a telodendria. I would ask you about the telodendria. But at the end of each of these branches, the tips of the axons get bigger. And they look like, they're called terminal knobs or boutons, since they look like buttons. Okay? Now, it is in these tips of axons that we find packages of chemical transmitters called neurotransmitters. All right, these are the chemicals the nerve cells produce and release onto other cells. Now, opposite the boutons on the next cell are channels for the chemicals that are being released from the tips of the axons. Okay? And so, because the tips of the axons release chemicals, opposite these will be chemically gated channels. So let me go back and put this in perspective. Let me repeat what I've said before. There are two major categories of channels. Those that leak because they don't have a way of closing, they don't have gates, versus those channels that are gated. That means that they have channels that can open and close. All right? Now, of the gated channels, of the channels that can open and close, the weight of the channels, what controls the opening and closing of the channels is what's next. That's the next division of the channels. Chemically gated channels are opened by chemicals called chemical transmitters, neurotransmitters. All right? What this illustration represents is the very end of an axon, where the axon forms a terminal knob or button or bouton. Inside of those boutons or buttons or terminal knobs is where the chemical transmitters are stored in vacuoles. Remember that another name for a small vacuole is to call it a vesicle. I thought it would be up here somewhere, but these would be called synaptic vesicles, but they're really vacuoles that contain the neurotransmitters. When the neurotransmitters are released, they will go across a space called the synaptic cleft that is between one neuron and the next neuron. And so it's because of this space that when the electrical part of the nerve signal reaches the end of the boutons, then the message cannot continue by these electrical changes. The electrical changes occur because of what happens in the membrane. And so there is no membrane bridging this space. But yet it will be chemicals that are released that will go across this space and influence the next cell to continue the signal or not. Okay, so we're looking at an enlargement of this space And opposite these boutons that are releasing the chemicals is where we find the chemically gated channels. However, it's on the axons of a neuron. I could be so bold as to use this drawing. Only on the axons of the neurons is where you find the voltage gated channels. Okay? The voltage gated channels, opening and closing, are controlled by the surrounding voltage, the surrounding charges. Whereas the chemically gated channels are opened according to the chemical transmitters that are released to them.
So would the voltage one be like a battery?
Exactly. Those charged particles are the same as how a battery works, especially a car battery.
And what about like the chemical one? What could you compare that to?
The chemicals that are released are just going to open the gates. But where the charge will come in is what kind of ions will be flowing through those gates. This is where the charge comes from. Okay, when we talk about, electrical charges flowing, it's the flowing of sodium ions and potassium ions and chloride ions that represent charged particles flowing. Okay, questions?
So they're the ones that keep the flow going. If there's no more of them to keep going, the flow, channel closes, or no? What manipulates the closing of the channel?
Okay, when there are two ways. When a certain amount flows through, the stimulus will open the channel, and then the chemical transmitter flows through. I guess it's like a fixed amount that flows through, and then the channel closes. This is a bad drawing. What I'm pointing to voltage gated channels, and what controls the closing, the opening and closing of these would be the overall electric charge around the channel. But when you get to the chemically gated channels like this, the chemical will go and bind to a receptor, and the channel will open. And now, when that chemical is removed from the receptor, the channel closes back up. What removes the chemical, we'll talk about, is what's called reuptake mechanisms. There are enzymes in the next membrane that will destroy the chemical transmitter, or the chemical transmitter will just diffuse away from its receptor. So that's how chemical channels will close. All right, I'll try to emphasize that again when we get to it, but at least repeat it again.